The overall goal of the following experiment is to follow the uptake of mitochondria into the vacuole as a means of studying the autophagy of mitochondria, a process known as mitophagy. This is achieved by expressing in yeast cells a genetically encoded and mitochondrially targeted dual-emission fluorescence pH-biosensor named MT-Rosella that labels mitochondria with red and green fluorescence. As a second step, yeast cells are subjected to nitrogen starvation in order to induce mitophagy. Next, we view cells by fluorescence microscopy or confocal laser scanning microscopy in order to visualize uptake of mitochondria into the vacuole. Results are obtained that show accumulation of red but not green fluorescence in the vacuoles of nitrogen-starved cells, indicative of mitophagy. Scoring of cells in a population allows the degree of mitophagy to be determined.
Using Rosella to monitor the uptake of mitochondria into the vacuole of yeast cells can provide mechanistic insights into the process of mitophagy. The approach can be adapted to monitor the autophagy of other cellular compartments, such as the nucleus. Demonstrating the procedure will be Dalibor Mijaljica, a postdoctoral researcher from our laboratory.
In this protocol, we use wild-type strain BY4741 and a deletion mutant strain delta ATG3 derived from the same genetic background. To visualize the localization of mitochondria in these strains, they are labeled with a fluorescent reporter, Rosella. Rosella is a dual-color emission biosensor comprising a relatively pH-stable red fluorescent protein.